Thanks for this opportunity to talk about one of the loves of my life, C4 grasses. So I won't tell you how long I've been working on them, but if you can dig my PhD out, you'll get an idea. So, in CABBI, we've chosen to focus entirely on C4 grasses, and those of you outside the plant community might wonder why. Obviously, usually if you talk to farmers or landowners about what trait they want in a crop, the first three traits they'll tell you are yield, yield, and yield. But of course, if they can minimize inputs and therefore costs, that's also an advantage. And what I want to tell you is why the C4 bioenergy crops we've focused on, the Andropogoneae, are particularly attractive in this respect. Now, first of all, why C4 photosynthesis? All carbon assimilated by terrestrial plants and nearly all algae has to go through one enzyme called rubisco. This enzyme evolved when there was no oxygen in the atmosphere, and it's actually labile to oxygen. So in the presence of oxygen, which of course there is for all terrestrial plants, there's an inefficiency in this enzyme. Probably every third step, instead of reacting with carbon dioxide, it reacts with oxygen. It burns off carbon, it's fixed, it wastes energy in doing that. Now, C4 photosynthesis, which has evolved at least 64 times independently, has found a solution to this. It's basically added on another layer of cells outside of the cells which contain rubisco, which serve as a CO2 concentrating mechanism. They pick up carbon dioxide with another enzyme, pepcarboxylase, put that into a C4 acid, which is transferred to the inner cells, where it's then decarboxylated to concentrate CO2 in these inner cells. At the high CO2 level of those inner cells, the oxygenation process is inhibited. And if you work through the theory, this makes C4 photosynthesis between 40 and 60% more efficient than the other form. By making it more efficient, it also allows the plant to fix CO2 with less water and less nitrogen. So traits that we very much want. Okay, that was a simplified version. This is actually the complete version in the group of plants that we're actually working at, because as I said, it's evolved 64 times. Each of those is a bit different, but this is the model which you has developed, which covers all of those reactions in those two cells. And we use this for then trying to re-optimize the process for current conditions. Back again. The most productive plant we know of on the planet is this Amazonian grass, Echinocloa polystachya. That produces almost 100 dry tons per hectare per year, and it's C4. The most productive temperate grass we know of is Miscanthus times giganteus. Sorry, Emily, this is your fourth showing this morning. Fifth if you count your run this morning as well. So this is Emily Heaton, stood by the first ever commercial plantation of Miscanthus giganteus in the United States. The highest productivity ever seen for that was 60 tons at one site in 1 year, but 30 tons on average is pretty average. When I first worked on this grass in England, we got 20 tons, and that is at 52 degrees north, about level with Hudson Bay. So another special feature of Miscanthus is its quite remarkable cold tolerance. So as I said, if we go through all the theoretical steps, we can come up with what the efficiency should be. And even at these high productivities, that's about 2% conversion of solar energy into biomass. So even these grasses are still not fulfilling the full potential of C4 photosynthesis. So this is showing sort of one description of the evolution of the grasses. And the box I put in red are where we see most of the C4 grasses. And that blue box is the Andropogoneae, which is the group we're looking at. And on the right-hand side there, you can see I've got another blue box, which is showing the grasses we're particularly interested in. Because this group contains miscanthus, sugarcane, sorghum, and maize, all quite closely related. And as we heard from Conchita, we think evolutionarily these probably separated around about 3 to 4 million years ago. So there's a huge amount of synteny in the genomes between miscanthus, sorghum, and sugarcane. And that is another reason for selecting these groups, because a genetic change you can make in one probably has value in all of the others. And of course, there's a great deal of synergy in looking at the genomes and their behavior. If we look at where these actually centers of origin, the center of origin for sorghum is thought to be in Eastern Africa, although there may be a second center in Western Africa. Sugarcane evolved in Papua New Guinea, although you can find saccharum species pretty well across the globe, although mainly in the tropics. Miscanthus is really hardly a cultivated crop. And again, miscanthus can be found in Africa, in India, through much of Asia. But we're particularly interested in the relatives of Miscanthus giganteus because this has these particularly high productivities, and its distribution, or its relative distribution, spread all the way from Papua New Guinea right up into southern Siberia. And indeed, this is work from Eric Sachs' lab. All of those dots are collections of Miscanthus saccharophloris, one of the parents of Miscanthus giganteus. And if you look at the hardiness zones it's covering, it goes right up into hardiness zone 3 in southern Siberia. And in fact, Eric has made collections from there. We've screened them photosynthetically. And after a long search, we found material which is even more cold tolerant than Miscanthus giganteus, which proved very hard to beat. Of course, one of the big advantages of C4 perennials, such as Miscanthus, you can see here is, because it's perennial, overwintering perennial, the same is true of semi-arid perennials. During the non-growing season, the nutrients are stored in the underground material. In the spring, that's mobilized into the shoots. In the fall, they're remobilized back into the root system. And so what you harvest is primarily, or should be primarily carbon. You can certainly select for that. And of course, this is a big difference we have with food crops, that we only want the carbon and hydrogen. We don't really want the nitrogen, which is so important for food crops. So there's a major opportunity there for selecting for low nitrogen. For Miscanthus in particular, but also true of sugarcane, is you plant once but harvest annually. You have minimal fertilizer use, minimal weed control. The side-by-side trials we did of Miscanthus and maize here in Champaign, we got 60% more biomass from the Miscanthus. That was not actually fertilized, although of course it is growing on a good Midwest soil. The perenniality allows, once weather conditions are good enough, for rapid growth in the spring. And so that, pretty well with Miscanthus, that eliminates weed control after the first year. The perennial binding the soil also is very important in stopping soil erosion, capturing any nutrients that might otherwise be washed away. And Evan DeLucia and Carbonacci have great evidence of that at the energy farm on instrumented field drains. You get rapid ground cover, which means you're intercepting radiation much earlier in the growing season, which was a big reason why miscanthus got 60% higher productivity. And there's net carbon deposition to the soil. As I mentioned, the genomes of all three crops have very high synteny. There's already been a large amount of resequencing. A very important aspect is their close relatedness means that hybrids can be made, and Eric Sachs has been successful in making sugarcane miscanthus hybrids, which mean we can hopefully move some of the beneficial properties of miscanthus, such as cold tolerance, into sugarcane. There is a large amount of genetic diversity. I mentioned lack of it in photosynthesis here, which I shouldn't have done because of Lisa's results. But what we have focused on is taking elite cultivars of sugarcane provided by Freddie, looking at can we now engineer in yet more diversity to get higher photosynthesis. Chris mentioned about vegetable oils from these plants. And of course, normally they produce very low quantities. But I should mention that Freddie and John have already managed to get 4.3% oil engineered into the stems of sugarcane. Now, sugarcane can produce 40 dry tons per hectare. If you could, or even more in some of the energy cane lines, if you can get that 4.3 there, that roughly means about 10 barrels of oil, compared to maybe 2 at the most from soybean. And certainly we don't think 4.3% is the end of that story. So the high productivity of these plants could allow large amounts of oil on some of that marginal ground, which we saw from a sustainability theme in the south of the US, and of course in Hawaii and Puerto Rico as well. Okay, so I'll finish there. Thank you.